all of this is to get a new product offering called metagenome-SIP, or metagenome stable isotope probing. And so this is a technique used to identify active groups of organisms within a microbial community. And this really touches on 3 milestones within the JGI strategic plan, which started with a pilot SIP viral metagenomics project, which then sort of paved the way for providing user access to metagenome SIP capabilities, which is what we're focusing on currently. And then shortly after, we'll eventually be able to provide user access to metatranscriptome SIP capabilities. And getting metagenome SIP going is also part of JGI strategy to address 3 VER grant challenges. So, of course, developing the metagenome SIP infrastructure at JGI, but then also, this will eventually couple metagenome data to biogeochemical processes, as well as functionally characterize microbial communities. And so the SI part of SIP is stable isotopes, and really we're focused on stable isotopes of these 3 elements: carbon, nitrogen, and oxygen. All 3 of these are found very abundantly in biomolecules, and all 3 of them have one isotope that is much more abundant than the others. So if we start with carbon-12, in natural samples, about 98.9% of carbon atoms are carbon-12, and then about 1.1% is carbon-13. So this carbon-13 has an extra neutron, which makes it slightly heavier. And then similar story for nitrogen-14, where most of it is found in the nitrogen-14 form, and then 0.4% as nitrogen-15. And oxygen has 3 stable isotopes, where most of it is oxygen-16, 0.2% oxygen-18, and then a very small amount of is oxygen-17. And so stable isotope probing is a type of experiment that takes advantage of these stable isotope abundances within biological material by incubate, so if you incubate microbial community or some culture, giving it substrate with the natural isotopic ratio, the active microbes will uptake that substrate and be able to incorporate those atoms into their biomolecules, whether it's DNA or RNA or protein. So in natural samples, it's mostly carbon-12. But if you imagine, you give microbes a labeled substrate, where something like glucose and all of the carbon-12 has been replaced by carbon-13. Microbes that are uptaking that substrate will now start to incorporate carbon-13 into their biomolecules, and they become isotopically enriched. And so there are various types of SIP to study all of these biomolecules, but in our case, we're really focusing on atoms that get incorporated into DNA, and a type of DNA, or a type of SIP called DNA SIP. And so at the very least, this tells us which microbes are actively taking up substrate and incorporating it into their biomass relative to others. Of course, SIP can also be a quite quantitative technique. So Bruce Hungate and colleagues developed a method of quantitative stable isotope probing for microbial ecology, or qSIP is what quantitative SIP is abbreviated to. And basically, for individual taxa, you can compare the change in density between unlabeled DNA. So this is DNA from microbes incubated in natural isotopic ratio media, and compare that to isotope labeled DNA. So DNA from microbes from enriched media, isotopically enriched media. And then the difference in the density between those DNA molecules is attributed to isotope incorporation. So DNA with more of the heavier isotope will be more dense, basically. And so we use this technique outlined in this paper and apply it to metagenome-assembled genomes. And another piece of the puzzle in our process of getting metagenome SIP going at JGI is using a high throughput system that automates some of the more traditionally tedious steps of CIP. Namely, a big part of CIP is collecting little droplets of DNA, and you end up for each CIP project having to collect hundreds of these droplets. And you can imagine if a person is doing that, it's going to take a lot of time, and that's prone to errors. And it's not something that can really be done at the JGI scale, where we're taking in hundreds of samples. And so in collaboration with JGI, Aaron Nuccio and colleagues developed this HT-CIP, or high throughput CIP, which uses robotics and barcoding in order to automate this very tedious step. And an added benefit of automation is better accuracy, or better reproducibility. So both of these plots are essentially showing the accuracy of collecting those droplets in the correct amounts and at the correct time. And so if we look at the left plot here, plot A, at the very top, that's when a person starts collecting those droplets, and you can see it's pretty consistent, since the density at which you're collecting droplets here is going to always pretty much start at the same, and it's kind of easy to collect the same amount of drops. But then as you continue along, errors start to propagate as you're collecting more and more drops, and the accuracy starts to get more spread out. But when you automate it, the robot is collecting these drops, and for the most part, it's a much more reproducible, much more accurate process, and so you get a much tighter grouping of automated buoyant density along this line here. And so combining the techniques from these two papers, JGI has developed a standardized quantitative analysis strategy for stable isotope probing and metagenomics. And basically, we have an analysis framework that is used to quantify isotopic enrichment on a per genome basis. So this is metagenome-assembled genomes using shotgun metagenomics. And so with these three papers, and along with all of the work of stable isotope probing that has come before it, they really laid out some strong footholds for getting us to the point where we want to be at, which is having a CIP analysis pipeline and data accessible to all. And so how do we get to that point? So it starts with the user having a sample of interest, and they perform incubations either with the control media. So this is media that has a natural isotope abundance. So most of the, let's say, carbon is carbon-12 in this control media. Or they do an incubation in an isotopically labeled or isotopically enriched media, where you're replacing carbon-12 with carbon-13. So this is, glucose with carbon-13. And then given enough time in that incubation, the microbes in the isotopically labeled media will start to incorporate that into their DNA. And then the user will do DNA extraction and send that to JGI. And then from there, JGI takes the sample, adds some pre-fractionation spike-ins, which are really for quality control. Essentially, when we get the sequencing data back, if those spike-ins are found in amounts or samples where they weren't supposed to be, we know that something went wrong along the way. But then those samples go through ultracentrifugation. And so this is an important part of CIP because isotopically labeled DNA, if it has the carbon-13, it will be slightly denser or slightly heavier, and it will work its way down the column further than isotopically light DNA. So you'll have the bottom portions of this tube have more carbon-13 DNA, and the top portions will have more carbon-12 DNA. And of course, it's not really a really steady band like is depicted in here. It's kind of a gradient as such, but you get the idea. And then once centrifugation is done, the previously tedious step of that was collecting those drops so that you collect different layers of this density gradient in a process called gradient fractionation. And so we generate about 15 to 20 fractions per sample. And so you get individual fractions ranging from isotopically light to isotopically heavy. And each one of those fractions goes through sequencing and data analysis. And a really important part of this for us is the addition of these post-fractionation spike-ins. And so these spike-ins really allow us to quantify DNA that is being mapped to bins that we generate from the sequencing data. And so the way we do that is during quality and contamination control, there's also a data report generated for those post-fractionation spike-ins, which gives us the coverage amount of them. Since we know the sequences of those spike-ins that are added, we can just simply map them, map the reads that are coming in to those sequences and generate coverages from each individual fraction. And then since we know the DNA concentration that we added to each one of those tubes, and we have coverage information, we can create a linear model where we equate coverage to DNA concentration. And then when we eventually go through assembly and binning, we get coverage information for each of those bins, where now we can estimate DNA concentration of those bins from their coverage values. And so a big part of this, and what we've really been working on a lot up until now, is the data organization of all of this. Because as you can imagine, is, if you have 10 samples and you have 20 fractions per each, you have 200 individual data sets that need to be labeled and grouped and assembled. And so the breakdown of this is, starts with the SIP proposal from the user, and the user will send in a certain number of source samples. So these are DNA that's been extracted either from control media or isotopically labeled media. And each one of these source samples is fractionated into some amount of fractions. And then on a per sample basis, all of the fractions within a sample go through a Metaspades co-assembly. And then we also do an all fraction for all samples co-assembly with MetaHitmer. And we don't do assembly of individual fractions because we found that any potential mag or bin data that comes out of individual fractions is largely redundant of what is found in these co-assemblies. And so the payoff of potentially finding useful information in individual fractions doesn't really equate to the cost of going through assembly and storing this data. So we only do these co-assemblies of samples and all fractions. And if you're assembling all of the fractions together, it ends up being a huge data set, and really something that's only possible with the use of MetaHitmer for many of these projects. And so MetaHitmer is a terabase scale assembler. And so metagenome-SIP is really leveraging a lot of the technologies developed in collaboration with JGI. And so each one of these co-assemblies goes through the standard annotation and binning pipelines that all metagenomes do. And some co-assemblies will have duplicate bins of other co-assemblies. So there's a dereplication process happening where you only keep the high quality bins, or the highest quality bins. And then we map reads from each individual fraction back to those bins. So we end up with coverage for each bin, we end up with coverage information from each individual fraction. And so this is, of course, a lot to keep track of. And so one of the things that we've set up with, thanks to Dana here, he really helped out, is an internal tracking method of source sample metadata. So there's this metagenome SIP source sample data warehouse report where it's searchable and filterable and really enables analysis to be done. as well as a metagenome-SIP fraction metadata warehouse report. And these two have been huge helps for me personally. And so this is great, of course, for internal use, but then for getting that data to the users, we also generate a data file which has a brief description of JGI Metagenome SIP, as well as a description of their proposal and the sample that they sent in, and then how that sample was fractionated and metadata within the fraction. And so that's, of course, useful for primary users who submitted the proposal and know what they're looking for. But we need to get to this data to secondary users. And so we've been coordinating with Gold, who they have started putting combined assembly SIP metagenomes on their platform here. There's 6 currently, and there's a handy help document to help orient users to this information, to this data. And we're starting to see metagenome SIP samples within IMG as well, and we're currently in the process of coordinating with them a way to visualize and distribute this data, since especially the combined SIP assemblies is a little more complicated than a standard metagenome would be. And so we're getting to the place that we want to get to eventually is where we have data available on Gold and IMG, and the data is being tracked internally and being distributed to users. And I'd say that we're pretty much at this point. And now the next step that we are getting very close to is running the qSIP analysis results or pipeline and getting those results to users. And eventually they'll have data like this, where they have a list of all of the mags that were generated from their data, or at least the high quality mags, and then how much isotope was incorporated into the DNA of those mags. And so getting to, I'd say this runs currently for mock data, and then I'd say the next step within the next month or two, we'll probably start getting this out to users that have submitted proposals. And then that will be probably what I talk about next time. And so thank you very much.